Just like mitosis was an evolutionary step that allowed eukaryotic cells to protect their nucleus integrity during cell division, meiosis is a process that allowed eukaryotic cells to perform sexual reproduction. In asexual reproduction, one parent cell becomes two new daughter cells, and they are basically identical copies of the original. But during meiosis, two different parents donate half of who they are to create a new organism that is more varied or is diverse. So sexual reproduction increases diversity and makes evolution explode in speed, and that's one of the main reasons that life has been so successful historically on Earth. And so certain kinds of eukaryotic cells in organisms, especially the multicellular ones, when they want to undergo sexual reproduction, they do the process of meiosis, where the point is to get a diploid cell that has two copies of each chromosome type or a pair, and make a half cell or a haploid cell which only has one chromosome of each type that we call a gamete. And throughout this process, at several points, it's going to generate more variation or diversity. So the whole point of meiosis is to make half cells so that new organisms across generations can preserve their chromosome numbers and at the same time create diversity. And that's the process that creates the gametes, such as the sperm and the egg that you see in the picture that we have there. And as you can see, it is actually a reduction division, where two division processes will create a reduction in the overall amount of DNA so that you can create those half cells that we were talking about. Not every cell in the body can undergo this process. Normal cells are called somatic cells, and they just basically undergo mitosis to be repaired, reproduce, or grow. But some special cells, called germ cells, have the capability of undergoing meiosis. And in some, these germ cells are in special locations called gonads. And these germ cells are the only ones which will undergo this process of gamete formation or gametogenesis. In humans, for example, this process happens because the adult multicellular organism undergoes a process of meiosis at their gonads, ovaries in females, testes in males, where the gametes are produced, egg and the sperm respectively. Now those are haploid cells which get together to form a new organism, which is a combination of both parents through a process that's called fertilization, which restores diploidy, creating a diploid zygote cell, which then undergoes mitosis and differentiation to develop into another adult multicellular organism which might find a pair, and we start the cycle again. And that's the cycle of reproduction, which is used throughout much of the animal kingdom. And the only difference, of course, is that different animals will have different karyotypes or different amounts of chromosomes inside each one of their cells. Humans, for example, have 23 pairs of chromosomes, 22 of which are autosomes, or the chromosomes which do the normal functions of the body and determine everything about who you are. The last pair, the 23rd pair, we call it the sex chromosomes. Now they do also some of the stuff that your normal body activities are supposed to be doing, but in addition to that, they also help determine your gender, especially the Y chromosome. Having the Y chromosome makes you be a male, and that's why males have 22 autosomes plus an X plus a Y, while females have a paired X chromosome for their sex chromosomes. Now notice that the karyotype chromosomes will have two of each type. One is going to be the maternal, or the ones that they receive from the mother, and one is going to be the paternal, or the ones they receive from the father. And so we are combinations of half of mom and half of dad. Notice that these chromosomes are only one single molecule or in a straight line, but that when division actually happens, these chromosomes will double up to the familiar X shape that you see, or the paired chromosomes, which are called the homologs, or a pair of a certain type, is going to be doubled into an X shape. By the way, the chromosomes are sorted in families, and they are also sorted by size from largest to smallest, with the 22nd pair being the smallest. Meiosis will start the very same way that mitosis will. It will start from a cell with an interphase, in this case a germ cell, that will undergo the S phase to become a cell that has a double amount of chromosomes. And basically, this, if the cell was the diploid 2N cell, at that point it was going to become a 2 times 2N cell. And then meiosis will start. And when you get to anaphase 1, the thing that's different that happens here that doesn't happen during mitosis is that those homolog pairs, or both copies of the same type. In other words, the chromosome 1 from mom and chromosome 1 from dad, which are the homolog pairs, will line up and form a tetrad, and then during anaphase 1 they will separate their separate ways. And then each of the daughter cells which come out at the end of meiosis will have only half the chromosome, because you actually separated the homologs, and that's unique to meiosis 1. And then in meiosis 2, those homologs which are now separated in different cells still have duplicates because they are sister chromatids paired up. And then the sister chromatids actually separate, creating a total of 4 daughter cells as the result of meiosis after anaphase 1 separates the sister chromatids. And so doing meiosis, meiosis 1 separates homologs, meiosis 2 separates sister chromatids. And now you are left with unreplicated chromosomes, or an end cell. Because after meiosis 1, what you actually have is a 2 times end cell. And then after meiosis 2, you have an end cell. Another interesting thing that will also happen during meiosis is the process of crossing over. Doing prophase 1, before you get to the metaphase, which lines up the tetrads and separates these homolog chromosomes during anaphase 1, doing prophase 1, those homolog chromosomes pair up with each other by attraction forces between areas of the chromosomes which are similar, and then they will actually become lined up in what is called a tetrad. And then the centromeres will connect and the tetrads will form, and then at certain points of those chromosomes will actually touch each other, forming a synapse. The actual location of each touch is called a chiasma or a chiasmata, and then something will happen at that point and crossing over will occur. Enzymes will cut pieces of each chromosome, corresponding pieces, and they will switch places. And therefore, at the end of prophase, you no longer have the maternal chromosome and the paternal chromosome. Each chromosome will actually be a mixture of both. So that once you undergo anaphase 1, and you see that at the bottom of the slide, you will see that each daughter cell will receive a chromosome that's not really the paternal or the maternal, but a combination of both. And that means that once you do the second meiosis and you do anaphase 2 and the sister chromatids separate, some of the chromosomes will be recombinations of the originals. And that's what we call recombinant chromosomes. This process, along with the separation of homologs that happens during anaphase 1, generates variability among the gamete cells and makes it more likely for us to have variety in sexual reproduction. And we'll review that again in a second. The process of meiosis actually includes processes which are similar to those that happen during mitosis. During prophase, the cell actually destroys its nucleus, it coils up, makes chromosomes, the spindle gets formed, and then you hit to metaphase where they're all lined up in the middle. But remember that during prophase, that chiasmata will actually take place, and the chromosome homolog pairs will pair up and form tetrads. You're going to have crossing over taking place. Then the crossed over chromosomes will line up at the metaphase plate, but not as they would line up doing mitosis. They actually line up as tetrads, which means doing anaphase 1, what separates is not the sister chromatids, but the homolog pairs, which sends one homolog pair one way and the other homolog pair the other way, which actually creates half cells. But remember, each one of these half cells still has chromosomes which are doubled copies because each chromosome has 2 sister chromatids in it, which means we need a second division to get them down to the chromatids. At the end of telophase 1, the cytokinesis will take place and the nucleus will come back to normal, but unlike mitosis, you don't go back to interphase and make another S phase. That would defeat the purpose of creating a reduction division. Instead, you go straight into meiosis 2, where now you have 2 cells separating because you had 2 daughter cells at the end of meiosis 1. So everything that happens will happen twice, one for each cell. Prophase, metaphase, anaphase, and telophase will repeat themselves, except this time, when the metaphase comes, it will be similar to what happens during mitosis, and the sister chromatids will separate to the opposite ends of the cell, creating 4 daughter cells which are all different from each other. And that is the process of meiosis. The interesting thing about meiosis, going back to the independent assortment of homologs that we talked about doing metaphase 1, you have different possibilities for the way these chromosomes can be lined up. For simplification purposes, we're going to look at a cell that has 2 types of chromosomes. And you'll see the first type listed on top and the second type listed on bottom. Also, for simplification purposes, deleting crossing over, just to keep things clearer. Imagine that crossing over doesn't even happen. You still create variation. Check it out. If on the possibility 1, you get all the paternal DNA in one side and all the maternal DNA on the other side. In other words, the part of you which comes from mom and the part of you that comes from dad go their separate ways during the anaphase 1. But since there's a 50-50 shot for each chromosome to end up in one way or the other, there's a chance that the exact opposite will take place, or that for each chromosome it will jumble it up. So notice that in the possibility 2, the paternal chromosome, the first one, went left, but the second one went right, and vice versa for the maternal chromosome. And so you create different combinations in that case. Notice that in the left side, you have 2 final daughter cells after meiosis 2, which are paternal-like, and 2 meiosis cells, which are maternal-like, on the left side. On the right side, with possibility 2 of a different kind of assortment during the metaphase 1, you're going to have a cell which has half mom and half dad. And then the other one will have a different half mom and a different half dad. So looking at only 2 different types of chromosomes, you have 4 different types of combinations that you could have possibly gotten from these 2 sets of chromosomes. Imagine something like human cells, where we have 23 different pairs. What are the chances that 2 cells, 2 daughter cells, will have exactly the same combination? Well, if you actually do the math, it will be 1 in millions. And so this is not going to be happening very often. Although, of course, in males you have millions of sperm produced every day, so it's bound to happen once in a while. But remember, you have to add crossing over to this. And so crossing over will jumble out the chromosomes even more and make it even less likely for 2 daughter cells to ever be the same. And so the whole point of this is that because of independent assortment of chromosomes, meiosis guarantees greater variability because it increases the chances of daughter cells or gametes which are different from each other, both because of independent assortment and because of crossing over. At the end of all of this, each of one of these daughter cells or gametes will combine with a gamete from a different person, which is usually random. Although you also have mate choice and all those different things happening, at the end of the day, a random gamete from dad and a random gamete from mom will collide and form a new child. And that is a lot of variation there. Independent assortment, crossing over, and random fertilization guarantees that sexual reproduction makes a lot of variation. So, that's why meiosis is so awesome, because it creates variation and it reduces the chromosome numbers in half, so that after every generation, you preserve the total number of chromosomes. Otherwise, you get a 2N cell plus a 2N cell and make a 4N cell after 1 generation. After 2, the process repeats itself. You get 4N cells and another 4N cell to make an 8N cell after 2 generations. 16, 32, 64. Eventually, the cell will be nothing but DNA inside of it. So, to maintain the number of chromosomes and to create variation, meiosis is the key, and that's why it evolved. Now, let's look at some differences between meiosis and mitosis. First, of course, the purpose is different because mitosis has to do with creating asexual reproduction or growth or repair, while meiosis is all to do with creating variation, maintaining the DNA count, and sexual reproduction. Both of them will start with DNA replication, but meiosis doesn't have a replication after meiosis 1. So that means meiosis 2 is the only type of cell division that is not preceded by a cell replication. During meiosis, there's 2 divisions. During mitosis, there's only 1 division. Meiosis will only happen in germ cells. Mitosis happens in somatic cells. Meiosis has 4 daughter cells, where mitosis has 2. Each daughter cell of meiosis is completely different from the other. In mitosis, they're clones of each other. The size is also going to be smaller for those gametes of meiosis since they underwent 2 divisions without interface in between, while mitosis tries to preserve the same size of the original cell. While one of them is better for copying, the other one is better for reduction and variability. Mitosis is what we call a conservative process, while meiosis is what we call a process that promotes variation. Meiosis obviously takes more energy since you're going to be making 2 divisions and extra processes, such as crossing over and independent assortment of homolog chromosomes. Mitosis make diploid daughter cells, while meiosis make haploid daughter cells from diploid mother cells in both cases. Now, the last thing, of course, is that problems during meiosis or mitosis can lead to genetic disorders, such as cancers and, for example, Down syndrome, where if one of those homologs fails to separate properly, or one of the sister chromatids fails to separate properly, you're going to create gametes with an abnormal number of chromosomes. We call that non-disjunction, in the process that actually creates a mutation called aneuploidy, or abnormal number of chromosomes. Now, normally you have only 1 chromosome pair that's actually affected, and you're going to have either more than usual, like in Down syndrome, 3 chromosomes 31, it's called trisomy, or 1 less than usual, what we call monosomy. But in case of plants, sometimes you have something that's called complete non-disjunction, or euploidy, or polyploidy, where entire sets of chromosomes get doubled, and that leads to the evolution of plants into different species as they become new karyotypes with multiple numbers of chromosomes of the same type. And that's basically meiosis in a nutshell, and I hope you learn a lot and don't do anything that would not make your mama proud.